in polymer chemistry at the University of Science and Technology of China. And he earned his PhD degree at the Institute of Neuroscience, Chinese Academy of Science. And he did his postdoc training at the Harvard Medical School. So he started his own lab in 2016 at the University of Michigan. So his research work has been focusing on the development, organization, and function of the spinal neural circuit mediating somatic sensation. And he has made several important discoveries in the field, such as the identification of the spinal cord neuron controlling and gating mechanical trend, and the spinal neuron mediating mechanical itch. So Dr. Duan's lab and my lab are working on very similar topics. So we work on somatic sensation, trend sensation, itch sensation too. But our lab are focusing on the peripheral sensory neuron, and Dr. Duan's lab is focusing on the spinal neural circuit. So I'm very excited to hear what he's going to talk about today.
Okay, so thank you for Liang's kind introduction. So I'm very happy to visit here at this moment. So compared to this very cold Michigan, it's the golden timing here. I'm coming here, so I'm happy to have met with some of the old friends, new friends, and I'm looking forward to meeting others in the afternoon. So today, the talk. So my talk, the title is the genetic dissection of the spinal circuits processing mechanical itch. So all the research interests focus on understanding how the sensory input to get a different motor output. We have this very fantastic sensory systems which can detect the different stimuli in the environment, including the visual stimuli, auditory stimuli, gustatory stimuli, the olfactory stimuli, and the somatosensory stimuli. Our brain, we can process those different sensory input through our thought, learning, logics, reasoning, and consciousness. Finally, we can get the different behaviors. So here, I just want to show you the very simple example to understand, to show you, to explain how the sensory input can get the different behavior output. This is very simple, the reflex arc system. But I know Tim will tell you, so it's not exactly the simple system. So this is the hand. We can touch the sharp needle. Those are the noxious, the stimuli can activate the mechanoreceptors, so in the nerve terminal, and then activate the sensory neurons in the dorsal root ganglia. This information processed to the spinal cord through the dorsal root, and then the information is processed in the spinal cord. Finally, activate the motor neurons in the ventral horn, and then can activate the muscle to get the withdrawal response. So those the reflex arc can help us to avoid the potential damage. So our question is how this spinal cord circuit can process this information, and finally we can get this motor output. We focus on the somatosensory system. Somatosensation is a sense to our body, including pain, itch, temperature sensation, touch, or body position. Here is mammalia's somatosensory system. Different sensory neurons are located in the dorsal root ganglia or trigeminal ganglia. They have a long axon processed to the different sensory organs, including skin, muscle, joint, or other deep tissues. Those sensory neurons can detect the different stimuli. For example, the C and A delta lung receptor, here I marked with red, can detect the noxious stimuli, including the proton, noxious heat, noxious cold, or noxious mechanical stimuli. And the A beta mechanoreceptor, here I marked with blue, can detect innocuous touch stimuli. Different type of information is processed in the spinal cord and then to the brain, can lead into different sensory modality. In the last decade, we are quite aware how the different sensory neurons detect the different stimuli. But how the information is processed in the spinal cord and the brain still remains a large unknown. So our lab, many focus on the spinal cord, This is the hemispinal structure. The neuron is a marker to label all types of neurons. So here, I just want to point out the basic feature of the spinal cord. The spinal cord is organized with laminar based on the morphology or intensity of the neuron location. In the dorsal horn of the spinal cord, this is the dorsal horn. This is the first station of the central nervous system received input from periphery. In the dorsal horn of the spinal cord, there are 5 different laminars. Projection neurons cover 5% of neurons in dorsal horn, located in the laminar 1 or deep laminar, which has a long axon project to the brain to transmit somatosensory information. Vast majority neurons in the dorsal horn are local interneurons, including 2/3 of exciter neurons, 1/3 inhibitor neurons. Those neurons are located from laminar 1 to laminar 5 to modulate the activation of projection neurons. The third part is different sensory terminal can project contaminant into the different laminars of dorsal horn. For instance, Lockshirt's C and A delta lung receptor predominantly project to the superficial of dorsal horn, especially laminar 1 to laminar 2. And a small portion can project to the deep laminar. Inoculars, A beta sensory input dominantly project to laminar 2 to 3 border area to laminar 5. So today I will tell you how the itching information is processed in the spinal cord. This is an ongoing project in our lab. So there are 2 forms of itch. One type of itch we call the chemical itch, announced the research, such as the mosquito biting. When mosquito bites the skin, we can trigger the mast cell degranulation and then release histamine. Histamine is the itching compound, can activate the receptor on the nerve terminal to generate the itching sensation. Finally, can cause scratching. So this type of itch we call a chemical itch. This induced by different the itching compound we call the pruritogens, including histamine, chloroquine, or other itching compounds such as serotonin. This type of itch is transmitted through high-threshold polymodal itching sensory neurons, as Liang identified MRGA3 positive sensory neurons in the dorsal root ganglia. This type of itch are histamine-dependent or histamine-independent. This is the animal model of a chemical itch. If we inject the histamine in the left skin, we can cause the scratching, the response of the hand paw towards to the injection site. We can count the scratching the boat to indicate the chemical itch responses in the animals. On the other hand, we have another type of itching form we call the mechanical itch. This type of itch is transmitted through the low-threshold mechanical receptor. Here is a human psychophysiological study published in 2013. In this study, the researchers used a very tiny metal loop to vibrate the venous hair close to the chin area. So those vibrations can cause intense itching sensation. This type of itch is histamine independent. That suggests it's different form of itch compared to the chemical itch. So here I just want to show the very simple experiment how we can trigger the chemical itch or mechanical itch. This is the chemo wipe, the tissue. I think most people have the lunch box. You can follow me. You can twist this chemo wipe, can form a very tiny tip, and then you can soak your lip area. You will feel itching. But if you use finger to cross your lip, you'll never get the itching sensation. How our system can modulate this mechanical stimuli can trigger the itching information. So this is our study. So on the other hand, so in the chronic itching patients, they have the common symptom we call the mechanical itch sensitization. That's because the gentle touch can evoke itching sensation on the normal skin. So these are 2 different types of the chronic itch patients. One is the atopic dermatitis. This type of patient suffer from skin inflammation. Besides the inflammatory side, other the normal skin, if you touch stimuli, can also trigger the extremely itching sensation. This patient even cannot put clothes on his arm because the gentle touch can trigger itching inflammation. Another example is the dry skin in older people. Those the dry skin patients always have the touch evoked itch. So how our system modulate or transmit those mechanical itch, the phenotype? So this is the question we want to answer in our lab. To learn from those the common processing of the somatosensory system. So this we call is the countersensory the stimulation, the effect. So here is a vision system. So middle is this the light. We can get the process, just like the location of the light. But the surrounding side have the counter light can suppress this the activation. And then we can get a very high fidelity stimuli in the vision system. In the somatosensory system, exactly the similar. So the stimuli can activate the sensory neuron in the middle, but the counter stimuli can suppress this information, the sensory stimuli in the surrounding side, and then can keep the high fidelity of this sensory transmission. So this is an example how we can use this process, this different the somato information. This means how touch can suppress the pain information. When you hit your finger, you always feel pain, but then you have to shake your hand. When you're shaking, the shaking hand can activate the A-beta mechanoreceptor in the joint and then can suppress pain. So 50 years ago, Petrevol and Zambazek postulated the very famous the modulation series. They call the gate control series to explain this counter information, the processing. They proposed the one type of neuron in the spinal cord. They call the pain transmission neuron. This type of neuron normally receive input from the noxious C and A delta nociceptors. When you hit your finger, the noxious mechanical stimuli can activate those sensory neurons and immediately activate the T neuron in the spinal cord, and then transmit information in the brain, leading to pain perception. But the T neuron also receive input from low threshold mechanoreceptor from touch fiber. But while normally touch stimuli cannot activate this neuron, instead they can suppress the pain information because of this kind of the feed-forward inhibition. The touch fiber can also activate one type of inhibitor neuron in spinal cord. Due to this feed-forward inhibition, so normally the touch stimuli can suppress pain. But in chronic pain patients, this inhibition is gone. The gate is open. And then the touch stimuli can trigger the pain information. We call the mechanical allodynia. Gentle touch can evoke pain. Let's go back to the itching pathway. So this is the itching pathway, process chemical itch. In the last decade, different labs, including our lab, identified the neural circuit to process this the chemical itch. It's similar to the gate control of mechanical pain. Indeed, they have the gate control of chemical itch. Zhou Chen's lab in WashU identified the GRPR neurons are essential to transmit chemical information. And Sarah Ross lab identified the B5 positive inhibitor neuron to suppress the itching transmission neuron to get chemical itch. Our lab and others lab identified the specific the pain neuron circuits can activate the B5 neuron to suppress itch. So that's the reason when you feel itch, you're always scratching your skin to suppress itch, because the noxious, the pain pathway can suppress itch. And Dr. Han Liang's lab did a very good the research to identify the MRGA3 positive, the sensory neurons is the itching transmission neurons in DRG. So this is the pathway to transmit the chemical itch. So what's the pathway to transmit the mechanical itch? Because the lightest touch can evoke itch, as I showed you before, but a little bit stronger the touch can suppress mechanical itch. How the spinal cord process mechanical itch information? We learn from the gate control theory of pain and the chemical itch pathway. We postulated the neural circuit to process mechanical itch in the spinal cord. In this are the working hypothesis. In this hypothesis, we proposed one type of exciter neuron. We call the mechanical itch transmission neuron. This type of neuron can receive input from the low threshold mechanoreceptor type 1. This is the mechanical itch transmission neuron in the periphery to transmit the light touch evoked itch. In the spinal cord, there is one type of inhibitor neuron we call the iron neuron here. Activation of this type of neuron can suppress the activation of the itching transmission neuron. This type of neuron normally receive input from another type low threshold mechanoreceptor receptor, which can mediate the touch evoked inhibition. So normally, this pathway is dominant on our skin, and we never feel out just the itching information in most of the skin. But in some of the chronic itching patients, this gate, the mechanism is gone, and then the gentle touch can trigger extremely itching information in our brain. So the question is, what's the cellular identity of T neuron in the spinal cord? What's the iron neuron in spinal cord to modulate the activation of T neuron? So today, I will talk about the three parts. Firstly, I want to show you, so mechanical itch is a distinct form of itch. In the second part is a published work. I will tell you, so spinal cord inhibitor neuron expressing neuropeptide Y can get mechanical itch. The third part is our unpublished work. We identified a unique type of spinal cord excitatory interneuron, which is the mechanical itchy transmission neuron. So as I mentioned before, the human is much easier to detect mechanical itch, but how can we measure mechanical itch in mice? So before our research, there's no any methods to measure acute mechanical itch in animals. So in our study, so we identified the behavioral assay to measure acute mechanical itch. If you look at the mouse, the skin areas, we found so the skin behind the ear is kind of different from other skin area. This skin area is much like the light, and it has a very the tiny hair there. After we shave the skin there, and then we use the van fiber to poke those areas, we can trigger scratching response. We believe this is a kind of the mechanical itch, the stimulation. This is a video to show you how we stimulate the animals to trigger scratching response. Use the van fiber to poke this area. We can always trigger scratching response there. We stimulate for 5 times and count the percentage of the responses, indicate this is the mechanical itch, the behavior. In this animal, they can always get a scratching. We call this the score is 100%. And then we use the local stimuli, use the 10 millinewton, the van fiber, to poke those areas, no any scratching. That indicate this can be mediated by the innocuous touch stimuli. However, when we poke other skin areas of animals, never get a scratching. This is example when we poke the lip area of the animal. He can feel something, but no scratching. This is we poke inside. That indicates in the most of the skin area, this stimuli is gated by inhibition. There's no any response in this animal. Furthermore, we found this kind of the itching is distinct from chemical itch. We use histamine antagonist to block the chemical itch pathway. We can dramatically reduce the chemical histamine-induced the itching scratching. But when we poke the same areas, the reduction of mechanical itch responses. That indicates it's histamine independent. Next, we ablate the spinal cord GRPR neuron. That is itching transmission neuron. For chemical itch, we can dramatically reduce the chloroquine-induced chemical itch responses. However, the mechanical itch keep intact. Suggest this is a distinct pathway to transmit the mechanical itch. Furthermore, we want to test what is itching the sensory neuron to transmit mechanical itch information. So here, so sodium channel 1.8 marks almost 60% of DRG neurons. We use the sodium channel 1.8 Cre to target a different type of the nociceptive cells, including cell nociceptor, cell itch neuron, including the MRGS3 positive neuron, or cell low threshold mechanoreceptors. And then we cross this 1.8 Cre with the Cre-dependent, the DTR reporter line. And then we can specifically express the DTR in the 1.8 pointed with the sensory neurons. This is the dipsorium toxin receptor. After we inject the dipsorium toxin, we can suppress the protein synthesis in those neurons, and then we can kill the cells. This example, we use the mRNA of 1.8 to indicate the successful ablation in the DRG. This is a control animal. We have this the robust, this is the 1.8 expression in the dorsal root ganglia. But in the ablated mice, we lost the most of this 1.8 signaling, indicated we ablated those neurons in the DRG. And then we test the itching behavior. We inject itching compound to trigger histamine-induced itch. We can almost abolish those kind of this chemical itch. However, mechanical itch still keep intact. That suggests those the 1.8, the sensory neurons are dispensable to transmit mechanical itch. It should be some other type of mechanosensitive fiber to transmit mechanical itch. So here I summarize all the first part. We identified so the unique type of this mechanoreceptor is A mechanoreceptor. We don't know is exactly the A delta and A beta. We'll tell you later. So which type of mechanoreceptor transmitted information? To transmit mechanical itch, distinct pathway to transmit chemical itch. So next question is, how does spinal circuit process mechanical itch? What's the mechanical itch transmission neuron? What's the inhibitor neuron to get mechanical itch pathways? To answer this question, so we try to dissect the spinal circuit that transmit and get mechanical itch. The overall strategy of our research try to introduce dipsorium toxin in the subset of spinal cord neurons, and then kill those cells to test to identify which type of neurons is the mechanical itch transmission neuron, which type of neurons get mechanical itch. So we learn from all the previous study from the autogeny of the neuron development of the spinal cord. In all the previous study, we identify the different markers which can label the different type of spinal cord neurons. This was different neuropeptide, different the proteins. We can label the different type of neuron in the dorsal spinal cord. But however, all of these markers are also expressed in other brain areas or non-neuronal tissues. How can we specifically express DTR in those, the subset of spinal cord neuron in the dorsal horn? Collaborating with Martin Golding at the Salk Institute, we developed those intersectional genetic strategy. In this strategy, we can specifically express DTR in the subset of spinal cord neurons. To achieve our goal, we did 3 different mouse lines. First, we developed the intersectional DTR line. The DTR expression is driven by pan-neuronal marker tau, but the expression of DTR is prevented by 2 stop cassettes. To remove the 2 stop cassettes, we did 2 different recombinase mouse lines. Here, we use the LBS1 Filippo to specifically express DTR in the dorsal horn of spinal cord. LBS1 is a transcription factor which controls the neuron development in the dorsal horn of spinal cord. Then third one, we use different the Cre line. We have identified those promoter by our previous the study. Combine these 3 different mouse lines together, we can specifically express the DTR in subset of dorsal horn neurons. And then we can perform the behavior screening to identify the neural circuits for mechanical itch. To evaluate our ablation, we cross the 4th mouse line together. This is Cre-dependent, the Rosa tomato reporter line. In the control mouse, without the Filippo expression, the Cre can trigger the fluorescence in the subset of spinal cord neurons, in the different exciter neurons or inhibitor neurons. In the ablated mice, we express DTR in subset of dorsal horn neurons. After toxin injection, we can selectively ablate those kind of the dorsal horn neurons, but not affect the expression of the neurons in the other type of neurons in dorsal horn or the expression in the brain areas. Using this strategy, we identified 2 different mouse lines, or 2 different neurons. One is the mechanical itch transmission neuron. One is the mechanical itch gating neuron. The next, I will briefly discuss all the published work. We identified the NPY neuron is a gatekeeper to prevent the mechanical itch transmission. MPY is a neuropeptide. Here we use MPY3 to target the MPY positive neurons in the dorsal horn. This is after it crossed with the reporter line. We found the MPY positive neurons are located in the lamina 2 to deep lamina. Based on our field mapping experiments, we identified vast majority of MPY neurons are inhibitor neurons. After our ablation, we can selectively ablate MPY neuron in the dorsal horn. This ablation did not affect the other type of neuron expression, such as the dilorphin positive inhibitor neuron, which can get mechanical pain. In our animal, 7 to 10 days after ablation, we detect those mechanical itch, the sensitization. In the control mice, if we poke the lip area, the liver gets the scratching response. But in the MPY neuroablated mice, when we poke the lip area, we can trigger robust scratching bolts. That indicates the sensitization of mechanical itch pathway. And eventually, this mice can develop spontaneous itch and skin lesion. We have to sacrifice this animal. Interestingly, ablating MPY neurons did not affect the chemical itch, the sensitization. In those animals, the different chemical compound can trigger the comparable responses compared to control mice, suggesting ablating MPY neurons did not affect the chemical itch processing. Moreover, in these animals, do not develop any the touch deficit or pain deficit, suggesting MPY neuron is selectively gets mechanical itch, but not other sensory modalities. Here we just summarize the second part. We identified the specific inhibitor neuron expressing neuropeptide Y. This type of neurons can get mechanical itch in the spinal cord. So next question is, which type of spinal cord neurons is the mechanical itch transmission neuron? So here, we screen the 12 different types of the excitatory neurons in the dorsal horn. And finally, we identified the eusensory neuron is the itching transmission neuron. This is the location of eusensory in the dorsal horn. After we ablate the eusensory neuron, we can selectively ablate this signaling in the dorsal horn, but not affect the eusensory neuron in the other brain areas, such as in the amygdala, which has been reported before. We can control the fear memory. So in the third part, I want to share all the ongoing project. We identified the spinal cord eusensory neurons transmit the mechanical itch. So this is firstly, I want to identify the laminar organization of eusensory neuron. Here we use different markers which can label the different lamina of spinal cord. This NK1R can mark the 80% projection neuron in lamina 1. We found most of eusensory neuron, almost all of the eusensory neuron tomatoes are located at the ventral side of NK1R zone. And none of these neurons overlap with NK1R, suggests it's not a projection neuron, it's local interneuron in lamina 2 to lamina 3. And then we use the different markers to identify the location of the eusensory neurons, identify this indeed located in the lamina 2 to lamina 3. This is the marker to label the lamina 1 to dorsal side of lamina 2 inner layer. This is a marker PKC gamma to label the 2 to 3 border area. And we also identified the 20% of the eusensory neurons are PKC gamma positive. This has been reported before. PKC gamma positive neuron dominantly receives input from a beta mechanoreceptor. That also suggests eusensory neurons could also receive input from a beta mechanoreceptor. And then we tested the neurotransmitter phenotype. We found the vast majority of the eusensory neurons express excitatory neuronal marker VT2, VGLUT2. And very few neurons express the inhibitor neuron marker, the PAX2. That suggests this is one type of excitatory interneuron, which are located in the lamina 2 to lamina 3. And then we test the mechanical itch behavior. We found in those neurons, if we poke the skin area behind the ear, no any scratching bolts induced. So this is a statistical analysis. The control mice, we can trigger the scratching bolts. But after ablate the eusensory neuron, which can dramatically reduce the scratching responses following the mechanical stimuli. After ablate another type of the spinal cord excitatory interneuron expressing the GRP, which have been reported for the chemical itch processing, there's any difference in the mechanical itch processing. That suggests this is a unique pathway, only transmit the mechanical itch information. More important, ablating eusensory neurons do not affect the chemical itch processing. Here we use the 4080 to trigger the histamine-dependent scratching response. No any difference in the control mice and the eusensory ablated mice. We also go through all of the different the sensory modality related behavior. In the eusensory ablated mice, there's low locomotion deficit, low touch deficit, low heat deficit, low mechanical pain deficit, low cooling or cold deficit. Also, we test the chronic pain the behaviors, no any difference in the control mice and those eusensory ablated mice. That suggests eusensory neurons is one selective, the specific part, the neuron circuit to transmit mechanical itch, but not other sensory modalities. Further, the study to underscore the function of eusensory neurons. Here we use acute silencing or activation the strategy to specific study the eusensory neuron function. This is the strategy identified recently. We use this the mutant of acetylcholine receptor. This have the endogenous ligand, the acetylcholine, so does not activate those receptors. Instead, the synthesized chemical CNO can activate those receptors. There are two forms of this mutant. One linked to the GI pathway to exert the inhibition function. One linked to the GS or GQ pathway to exert excitation the mechanism. Here we use intersectional strategy to specifically target the spinal cord eusensory neurons to express either the inhibitory, the dread receptor, or excitatory dread receptor. Here, we silence eusensory neuron in the spinal cord. In the control animal, when we poke those areas, we can trigger the scratching bolts. But after the animal inject the CNO, when we poke those areas, no any scratching bolts. That indicates after we silence eusensory neurons, we can suppress mechanical itch induction. More interestingly, when we directly activate eusensory neuron in spinal cord, we can trigger spontaneous scratching. As indicated, this type of neuron indeed can trigger the scratching behavior, the motor output. So all the ablation, silencing, and activation behavior assays underscore the function of eusensory neurons as mechanical itching transmission neuron to transmit mechanical itch. So next question is, so how the spinal cord eusensory neurons process the mechanical information to generate this mechanical itch? To answer this question, we use the spinal cord slice recording to identify the selective transmission of eusensory neurons in the spinal cord. So firstly, we try to identify the different stimulation from periphery. So use this, contain the spinal cord with the dorsal root. We can identify different stimuli by the electric stimulations. If we use the high intensity, the electric stimuli, we can activate the A beta, A delta, and C components of the dorsal root responses. If we reduce the stimulation intensity to 25 microampere, we can specifically activate the A beta fiber alone. Do not activate the A delta or C fiber. Use this strategy, we can identify the sensory input which can project onto eusensory neuron in the spinal cord. These are the spinal cord slice recording the system. We use this the sagittal spinal cord slice, the preparation which contain the DRG and hold the dorsal root. We use this the suction electrode. We can stimulate electrically the dorsal root to activate the different type of fibers. And then we can detect the septal transmission which can project onto the different type of neuron in spinal cord. Here we record the eusensory tomato positive neurons in spinal cord slice. We detect vast majority of eusensory neurons in laminar 2 to 3 border areas receive the A beta input. And very few eusensory neurons in this area receive input from A delta fiber. And none of these neurons in the 2 to 3 border area receives input from C fiber. Our study suggests eusensory neurons receive dominantly from A beta mechanoreceptor. And interesting, when we record those UCN3 neurons, look at this, the diagram. We record UCN3 neuron here. If UCN3 neuron receive this feed-forward inhibition, so normally this pathway, the A-beta stimuli, could be gated by this inhibition. Indeed, when we record UCN3 neurons, we can record both the excitatory post-septic current and the much stronger inhibition there. Because of this inhibition, when we use the current cam, record this action potential induction, normally A-beta stimuli cannot generate any action potential in the UCN3 neuron. Instead, we can record much stronger the IPSP on those neurons. When we remove the inhibition by bus application the bicalculin strychnine to remove this type of inhibition, the same stimuli can trigger robust action potential induction in most of UCN3 neurons. This study suggests UCN3 neuron dominantly receive input from A-beta mechanoreceptor, but normally this type of excitation is gated by inhibition. If we remove inhibition, the gate is open, and then A-beta stimuli can activate the UCN3 neuron to trigger mechanical itch, the induction. So what's the inhibition? Is this inhibition come from the MPY neuron? To address this question, so here we use this intersectional, the rabies virus tracing strategy to specifically identify the preceptive neurons which can project onto UCN3 neuron. In this strategy, so we express this is the histone GFP. We can mark this is the Cre positive neuron, Cre and Filippo positive neuron in the dorsal horn. And then this manipulation can also express this we call the TVA receptor. It can bind this is the envelope protein from rabies virus. And also we have this the G protein of the virus. This intersection, the rabies virus, the mouse tools, will allow us to specifically trace the preceptive neurons of this the target neuron in the dorsal horn. We call this one is the starter neurons, express Cre, Filippo, and HTP. This neuron is marked by green. Here is local the neurons, infected neurons, because the expression of TVA on the membrane, and this allows these neurons can bind this is the pseudotyped the rabies virus. And then so the pseudotyped rabies virus, which deletes the G protein normally, can package the G protein in this cells, in the starter cells, and then can infect the preceptive neurons. So starter cell labeled by the M cherry, the red and the green, and the preceptive neurons labeled by the red only. In our study, we identified this is the one is the starter UCN3 neuron in spinal cord, and then we can detect the multiple the red neuron surrounding those the starter cells. That indicate those the red neurons are preceptive neurons which can project to those neurons. And we also identified some of those neurons are MPY positive. That indicate those MPY positive inhibitor neurons can form this monoseptic connection which can project to UCN3 neuron to get UCN3 neuron. So this is our conclusion. So we identified so mechanical itch is a unique form of itch. Spinal cord MPY inhibitor neurons can get mechanical itch, and the spinal cord UCN3 neurons transmit mechanical itch. So we still have no ideas which type of projection neurons can project this is the mechanical itch information to the brain, and which type of A-beta mechanoreceptor can activate the mechanical itch pathway, and which type of A-beta fibers can suppress this mechanical itch. So here is all the labs in our lab. We have the two postdocs. So Hani did all of this the recording in the lab, and other people contribute to the behavior or other assays. And I would like to thank my collaborators or some of the previous the mentors, Qiu Fu, and the Martin Golding offers this the ABS1 Filippo, this intersection of the DTR and intersection of HTD lines. I thank the Shu Daimiki to offer this intersection of dread mouse lines. We thank the Zhigang He to help us for those the rabies virus tracing at the beginning. So we always would like to thank the GeneSat program and NIH and Annenberg Atlas to provide some of the very fantastic mouse tools to allow us to target the different neural circuits. So tonight, so is the NCAA the championship. So here I would say the go blue. Okay, thank you.
set of sensation. With that being the case, it's easy for me to understand, so I'm not from this research area at all, it's easy for me to understand why we would still want a separate channel for chemical itch. But what's the evolutionary value of a separate mechanical itch processing stream that sort of operates independently from touch and pain? It seems like those are the things that really worry about.
Yeah.
If something touching me, I want to find out what it is. Is it dangerous? Obviously, if it hurts. But what is itch independent of those things?
So the question is from evolution point of view. So how this different pathway are separated, right? So here actually, so we have no the exact answer, but so we have some examples, like the animal. If you have the insect, right? Some of the dangerous insect touch your skin, you can trigger the uncomfortable or itching information. We call this is the mechanical itch pathway, and the sensation can allow us to avoid the potential, like the biting of the dangerous insect. Actually, this is from the evolution point of view. This is the significance of this, the importance of the mechanical itch sensation.
But is that something we don't get from touch?
So no. So normally the touch, so we generate most help us to identify the different material. Even some people also reported before, the touch can help us to get some of the pleasant sensation, emotional component. But the touch normally is not can transmit those uncomfortable information. So here we have the mechanical itch pathway and help the animal, help a human to detect the uncomfortable stimuli and avoid the potential, the damage in it. Okay, yeah.
So in amputation, a big problem for patients is that they experience phantom sensations, and one of those sensations that's pretty common is a phantom itch without, obviously, a peripheral stimuli because
Yeah.
there is no lid there. So where do you think that might fit into your pathway? Like what light can you shed on maybe where that pathway is breaking down in amputation?
Yeah, so as I discussed at the beginning, so our research interests focusing on understanding how the sensory input to get the motor output. We focus on the sensory input. So most of all the current, the pathway focus on we have to receive the sensory input and then can generate different perception. For the phantom pain or phantom itch, like the recently identified by Zhou Feng's lab, they discuss the contagious itch. When animal watch other animals scratching, this animal can also feel itch. We call it the psychological itch. It's directly come from the brain. So that's without any the sensory input. Our brain receive input from the vision system still can get those itching sensation. This is different from our current study. We focus on somatosensory system.
But they still have an intact all the spinal pathways for.
That's no spinal pathway. That's a phantom, the phantom itch.
That pathway is still without the peripheral input. The rest of the circuitry is still there. So you don't think that could be spinal at all?
Yeah, that's not from spinal cord. So spinal cord receive the somatosensory input from skin, from other areas. And then all have the projection neuron pathway go to the brain. Finally, we can generate this itching perception in the brain. But that's the phantom itch or contagious itch can directly come in the brain. Receive input from the vision system and then can trigger some of the psychological, the interaction between the vision system and all the itching center in the brain could cause those perception.
Kind of a follow-up on that. It seems like maybe one benefit of having separate channels and lots of potential intermediate cellular subtypes is you can have descending modulation in multiple ways. So have you thought about how the descending pathways might be gating the mechanical control?
Yeah, that's a good question. So now, we use the similar strategy, the intersectional strategy. We start from the spinal cord using neurons. We try to identify those descending inputs in the brain. Can we find out some like the neurons in the brain which can, through the descending regulation, to modulate this pathway in the spinal cord? Yeah.
So there are descending pathways, and is that modulated? Because I was thinking when I was watching the videos of the animal being stimulated, they have visual the stimulator also.
Mhm.
I'm wondering to what extent.
Maybe can suppress or maybe something regulate. Exactly, yeah, it's possible. So like the attention can always suppress pain. But when animal will concentrate to watch something, they can have low, such as the pain sensation. Exactly, I guess in this pathway could be have the similar regulation from the brain system. So, but we don't know. So which type of neuron can receive those descending the regulation? So now we try to start from this one, and then maybe later we can identify which is the projection neuron, and then we can think about so what's the descending regulation on all the mechanical itch pathway to regulate those the itching the information in spinal cord. Yeah.
Do you think your control of the itch perception is? It seems like it's either itch or no itch.
Mhm.
Do you think you have good control of the degree of itch?
So in the normal or physiological conditions, there's no kind of this degree. Mostly it's just stimulate and get a response. But I believe in some of the chronic itch, the models, we can find out the different level of just the scratching or some like the mechanical itch sensitization. And then, yeah, we are also testing the difference, the chronic itch model to identify the pathway's function, how this pathway the dysfunction of this pathway can cause those mechanical itch sensitization on the different chronic itch models. Yeah, we are testing that part.
I'm just thinking from
Yeah, exactly. Exactly. Yeah.
Well, it's not worth the trouble.
Exactly. Yeah. You can look at there's so many the different synapses. It could be it occurred, right? The different level of the regulation can modulate the behavior output. Definitely. Yeah. Yeah.
When you use the triad system to activate the UNC3 neuron, you show the animal have spontaneous eating.
Mhm.
Right?
Yeah.
So which specific area do they scratch? Do they just scratch the ear?
Not just the ear. But this is also very interesting. I guess, so firstly, why the animal uses the handpaw to scratching? Because handpaw can get much stronger mechanical stimuli than the forelimb. The forelimb, the wiping, is kind of the touch information can suppress pain. It has been reported before. But animal use a handpaw. So normally when we use a handpaw to scratching, they kind of scratch in lower back. They have to use the mouth, the teeth to bite this area. But it's hard to detect. But the handpaw mostly has scratching the toes to the area close to the head. It's much easy, right? Animal much easier to scratching. So that's the reason when you activate those different itching the pathways, you can always get the spontaneous scratching with toes to the head area or lap area, because for animal it's feel more comfortable.
So I just think it's very interesting that that's behind the ear. That's the only spot that you can induce.
You can induce that part, because why? So only the area can get the mechanical itch. So we are collecting some of the sample in this area, want to detect what's the difference between this skin area and other skin areas. So yeah, it's an interesting question. So our hypothesis could be one, maybe have much the enriched the mechanical itch fibers there, but we don't know what's that fiber currently. The second, maybe very few the inhibition pathway, very few those fibers there. It's both happen, yeah.
Thanks very much for coming. And thanks, Dr. Duan, for the nice seminar.
Okay, thank you.